everyone. Let's start from where we left in the previous class. We were discussing how two component systems are of different types, canonical and hybrid one, and we also ended up terming hybrid two component system as a three component system because it needed another component in between to mediate the phosphor transfer or phosphor relay. We saw example of how bacteria undergoes different life forms utilizing this hybrid two component system. We will see more examples today that how bacteria needs to communicate to its environment and responds in a very proper manner by utilizing different two component system, whether they are hybrid type or canonical type. So just a recall that simply you have two types of two component system, hybrid and this. Here you have the sensor kinase, which will have only histidine which is eligible to receive the phosphate group. In case of hybrid, you will not only have the histidine but also have the aspartate residue which can mediate the phosphor transfer. This will require the next molecule as a response regulator, which will have the aspartate residue and accepts this phosphate group on it. However, here you will need additional molecule which we called a minimal histidine containing domain containing protein, which is a histidine phosphotransferase. So this phosphate is transferred here, and eventually from here you will have the aspartate residue going on to the response regulator. We also understood that how a series of response regulators could be working together to ensure that a response is met, basically in response to the signal which is present outside or something which is generated locally inside. The response regulator must get into action. So today we will cover a couple of examples that how the two component system is working tirelessly to ensure that bacteria is in equilibrium with its environment. So we will take example of chemotaxis here, and this chemotaxis will be inducing movement of bacteria towards or away from the source from where the signal is coming. We all understand that if we like something, we move in that direction. If we don't like something, we move away from that direction. Same applies to bacteria. If there is a odor, there is a source of chemical which bacteria is liking, it will rapidly move in that direction, and if it does not, it will move away from that direction. Also, bacteria will need a lot of flagellar movement. The surface of bacteria protruding this flagella, their beating in a given direction allows movement towards the chemical or moving it away from the chemical. You can understand that clockwise movement, when it happens, it will actually take it towards the bacteria. The clockwise movement of the flagella will take it in the direction where signal is coming from, and anti-clockwise movement will kind of backpedal. So bacteria responds to extracellular nutrient, small molecules and ions in terms of chemotaxis, and accordingly they exhibit the chemotaxis behavior to these environmental cues. So let's see how such a system of two component system is operating here. Think about a scenario where the bacteria is in a milieu where this is the plasma membrane or cell membrane. In this particular scenario, the two component system sensor kinase is something which is not directly sensing the presence of Q. This molecule which is shown here, this is basically an adapter. Through its extracellular domain, it will sense the presence of small molecules, and once the small molecule is occupied here, this the receptor or the adapter should undergo some change. And one of the things which changes with this receptor is the methylation. And how that dynamics of methylation, whether it is methylated state or demethylated state, is helping respond bacteria to this chemotaxis event. So the important molecule here, you will find that there are multiple of these proteins which are named like A, Y, B, W and R. These are the components of the two component system which bind to the receptor or bind to this adapter and help mediate the response. So small molecules, whether they are amino acid derivatives or some sugar, they actually trigger this adapter molecule. So what you see here as a component A, this is the kinase. What you see as R and these Y and B, they are response regulator. And since this adapter, which is membrane bound, needs tethering of this kinase, you have this molecule W, which will be ensuring that the kinase is tethered. So if you look individually those components, this is what I was telling you, the receptor, it has the extracellular domain, receptor or adapter, whatever you wish to understand this as, just that it should not be confused with the sensor kinase or receptor histidine kinase. It is not the kinase domain containing receptor. It is in that sense an adapter molecule which allows the kinase to come and dock and perform the signaling. So this receptor has a ligand binding domain to which it will bind to different types of cues. And internally it has the domain which binds to Che. Here you will see that these molecules having a prefix Che or K for chemotaxis. So the kinase here is Che if I say, and this must bind to the receptor. That's why the intracellular domain of the receptor has Che binding domain. We will also see that Che W must also bind to the receptor. Why? Because this is the one which holds Che to the adapter receptor molecule. So both of them should have a binding space on the receptor. Also, I told you that this receptor is regulated by a methylation event, and that will be performed by this molecule which is CHER, so that is also having a space for binding. Your actual sensor kinase or histidine kinase is this Che molecule. So it has a histidine residue at position 48, which will receive the phosphate group as the signaling will proceed. It also has this dimerization and catalytic active domain, which basically means that it will allow the phosphorylation of histidine residue. Also, since it is internal molecule, it has to give signal to the other molecule response regulator. So this Che Y and B, they should be binding to this kinase. And this Che W protein, which was tethering the Che to the receptor, that should also bind to this molecule. So this picture represents that what are the basic units which are present in the molecule and where are those interactions which help it perform in the chemotaxis two component system. So this was your kinase, and one of the response regulators is Che B. What you realize that it has a minimal aspartate residue where phosphate will be moving, and it has some catalytic activity, methyl transferase domain, right. So it will also be affecting the methylation status of the receptor. Okay. So this is not the basal type of response regulator which has only the receiver domain, but this is more complex type of response regulator which has additional catalytic activity of methyl transferase type. The other component of this signaling TCS is that it has a CHER molecule. This is also a response regulator which affects the methylation status or it promotes the methylation, and then you have the receptor binding domain. Remember, this is which was directly binding to the receptor. Then you have a simpler molecule, a simpler response regulator, which does not have additional domain, but it has only the aspartate residue where phosphorylation can happen. So you will find that there are multiple of these molecules which are present over there, and they play crucial role in ensuring that the movement of the signaling event is in forward direction, giving the feedback signals so that the receptor activity can be regulated, and also ensuring that different responses are mounted. Remember that this is a chemotaxis kind of response, so bacteria, depending on the signal, may have to move in the direction where the signal is coming from, or it has to move away from the signal. And for that, we remember that we discussed that flagella rotation is very important. And for that, you have these molecules which are in the film category. There are different types of film. There are basically 2-3 different types. They form the heterodimeric trimeric complex, which affects the flagellar movement. Then you have this Che-G molecule, which is a regulatory molecule. So what appeared in the beginning to be a simpler, or what I told you earlier that okay, 2-component system seems to be very simple, but what you're finding here is that it's very complex, right? It just tells you that how diverse these processes pathways can be. By integrating multiple different factors, you exert more tight control on the signaling. So if you just summarize all these molecules and put them inside the cell and see what happens. So how exactly the system will be initiated? The receptor senses small molecule. Consider that it senses sugar, and bacteria will love sugar and move in that direction, right? So once this receptor is activated because of the binding of this, it will help recruit this Che-A, which was your kinase. And you remember, kinase was tethered to the receptor with help of this Che-W molecule. So Che-W is not a response regulator. Why? Because it does not have that conserved aspartate residue. Rather, it's a adapter kind of molecule which tethers kinase to the receptor. Once this kinase is tethered to the receptor, it will be activated because of autophosphorylation on the histidine residue, and that will divert the signaling forward. So one of the simplest thing which can happen in response to that is the flagellar movement, so that bacteria moves in the desired direction. So this kinase will transfer the phosphate group on the Che-Y. Che-Y will work through the film-like molecule, which will ensure that the flagellar movement is activated. This molecule serves as a phosphatase. So in some sense, internally the system is regulated by phosphorylation or phosphotransfer on Che-Y by Che-A and dephosphorylation of Che-Y by Che-Z. Together, the levels of phosphorylated Che-Y will activate this flagellar system for the bacterial movement. This is one scenario. How other receptor or other response regulator works, that this particular response regulator, if I can just label simply for the sake of understanding this as a response regulator 2 and this as a response regulator 1. So this response regulator 1 mediated the movement of bacteria towards the source. The other system which works through Che-B. When the phosphotransfer happens to the Che-B, this is activated, and what it does is that it removes the methyl group. In addition, this Che-R, which is here, what it does, it adds the methyl group to the receptor. So the dynamics of methylation or methyl plus versus methyl minus, where this methylation is happening, it's on the receptor. So the dynamics of receptor in terms of availability of methyl group to its sequence or removal of the methyl group, keeps it into the active-inactive mode and keep responding through this Cha system. So this is also a feedback regulation by modulating the levels of methyl attached to the receptor. And the other feedback or the system which is here, where Cha activates Chy, which activates the flagellar system. And how this is internally regulated is phosphatase activity, right? So basically, in response to the chemical, or here we took an example of sugar, it will allow the movement towards the source of sugar, and you'll have clockwise movement so that it moves forward, and then you will have the bacteria responding to the chemical cue in its surrounding. So you realize that how complex set of compounds which are present, or the moieties which are present like Chr, Chy, Chz and phlegm, they regulate the movement of the bacteria, activation of the receptor or the adapter, and proper response to the chemotaxis for small molecules like sugar or amino acids and so on. If we take another example of where two component system is working, is about the life form fate. Some bacteria, they do exist in different life forms, in the sense as a free living and tethered to something. We saw in biofilm, they were tethered to something, making a biofilm. This situation is different because here the cue to remain swarmer or sessile is very different. So this bacteria exists in the life form, different life forms, which is swarmer or sessile, meaning here it will be freely moving, utilizing its flagella, moving from one place to another, and this will be tethered to the substratum. So the sessile form will be tethered to substratum. So basically, only when this bacteria is tethered to the substratum, it can divide. Otherwise, it will be freely growing and moving. So this bacteria has different cues: cell cycle cues, whether to divide or not, and cell type cues, whether it is swarmer or sessile type. Here you involve multiple different types of two component system to help mediate this process. So one of the two component system which you utilize here involves div-J and div-K, and the other two component system which works here is CCK-A and CTR-R. Active div-K induces div-J and inhibits the phosphatase activity. We will see in the coming slide. So basically, you will have one component system which will be ensuring that the bacteria is dividing or cell cycle genes are regulated, and the other thing which will be regulating the cell fate. And we will realize that this one of the two component system is the canonical type and the other one is hybrid type. So let's see these components, how they come into picture. So consider a situation where this bacteria is growing. You might have heard that bacteria generally lives freely as long as nutrition is available, there is no scarcity of that, and there is no external pressure. They undergo sporulation. Some bacteria undergo sporulation, which actually talks about the environmental condition. Similarly, here this bacteria, Colobacter crescentus, has to decide at some point of time to divide or keep moving as a free form. So there will be cell cycle cues which will say that okay, you probably should go under division or not undergo division, and there will be cell type cues which will be telling that okay, you remain as a swarmer, enjoy life that way, you don't need to undergo the division. So the first set of this two component system works through div-J, which is your sensor kinase, and it directly works on this response regulator which is div-K. So when there are cell type cues, div-J signals and activates div-K, and it remains at one of those type, whether swarmer or sessile. We will see more of that. Then there is a cell cycle cue which says you should undergo division. that induces a molecule which is PLAC, which is a phosphatase, which will inhibit DIV-K as a response regulator. So there are parallel arrangement from different cues. One tells that okay, this molecule should be activated and identify itself to a particular cell type. Other system will work through and say that okay, you need to undergo cell division. So this is one two-component system working when it comes to responding to the division or free living form of the bacteria. So the other kinase or the two-component system which works here is working through the hybrid kinase CCKA and the master response regulator CTRA. Since this is a hybrid kinase, it also involves the histidine phosphotransferase, which is this molecule. So normally what happens that when there is a cell type Q, meaning a bacteria is preferring to remain as a swarmer bacteria, it will activate DIV-K, and this inhibits the other type of histidine this inhibits the other type of two-component system. So basically, as long as cell type Qs are pronounced, there will be no cell division, or it will not undergo cell cycle division. So first system inhibits the other one. In situation where this thing is inhibited, DIV-K is inhibited. DIV-K, when I say, it's a phosphorylated DIV-K which is inhibited. How that will be inhibited? If for given condition cell cycle Qs overwhelm, they activate the phosphatase and remove this phosphate group from DIV-K, which makes it inactive. When this is inactive, this inhibition is lost. Now this kinase is ready to function. So when this kinase is activated by a phosphorylation event, which involves its own set of mechanism, it will need the histidine phosphotransferase because it is a hybrid type of kinase. Now this histidine phosphotransferase will transfer the phosphate group to the master response regulator, and this will be serving to activate cell cycle genes. Basically, it will have those transcription factor kind of activity to induce the cell cycle genes. So let's see what all is going on in this scenario. So there are two sulfate which this bacteria has, stalked sulfate and swarmer sulfate. Let's identify them with the different design. So this is your swarmer sulfate and this is stalked sulfate. So what you notice here is that you have one situation where there is a signaling composition of DIV-K and DIV-J. What happens in case when it starts to become a stalked cell type? High DIV-J activity is there, and you will see that it is localized to the substratum. Now what will go on is that cell, when it becomes attached to the substratum, it decides to divide. Basically that is one of the first call it has to take before it goes and attaches to the substratum. Here if the cell is in G1 phase, this is more like S phase, G2 and M phase type. If I say M phase, bacteria do not have M phase, they generally just divide, right? So once the bacteria decides to undergo the stalked sulfate, it will attach to the substratum. You will have activity of the DIV-J, DIV-K. You will activate the CCKA kinase, and you will ensure that you will have more of the dephosphorylated molecule, and eventually what will happen that once these Qs are set in, the bacteria elongates, becomes bigger, transcription of those cell cycle genes come into play, and then the division happens. And the cell which is not tethered to the substratum becomes the swarmer cell. And one of those type of signaling will be going on. Here you will have div J K kind of signaling. Here you will have C C K A kind of signaling going on. So what you understand here is that the two different kinases or two component systems are working together to tightly regulate whether the bacteria should attach to substratum and take a decision to divide and give rise to the new set of free swarming bacteria, or depending on the other two component system working, the system keeps on dividing, the system keeps into the swarmer fate. It does not take a call to attach to substratum and to divide any further. So what we have understood from this class is that the two component system is important for multiple different functions where bacteria responds to the environment. It could be a chemotaxis, whether the sugar is present or amino acid is present or any other small molecule to which that adapter was responding to. And then there could be multiple different two component systems which will work together. In case of chemotaxis, we saw only one two component system working and the kinase there was Cha. But in case of this sessile, or I should say swarmer versus stalked cell fate, there were two different types of two component system working, one which was with div J and K and the other one was CCKA and CTRA kind of combination. The kinase and response regulator, same here as a kinase and response regulator. So what we can say in nutshell, that two component system is required for vital functions which help bacteria mediate its responses to the environment and survive in the given environment in the most efficient way. Thank you.